Wastewater is the most essential element of life. The United Nations Sustainable Development Goal 6 is to ensure the availability and sustainable management of water and sanitation for all. Specifically, Goal 6.3 focused on improving the water quality, wastewater treatment, and safe reuse for people globally. It also aims to achieve a reduction in pollution from the release of hazardous chemicals into water sources and the ability to treat, then reuse water sources, a key aspect in conserving limited water resources. A cornerstone to the realisation of the goal lays in the development and implementation of biological treatment processes, particularly with the use of microbiology to remove nitrogen and phosphate and other pollutants from unsafe and unusable water.
Photocatalytic detoxification and nanotechnology filtration are two innovative approaches that have shown significant success in improving water quality. Photocatalysis, which involves utilizing titanium dioxide activated by UV or solar light, was a method primarily tested in laboratory settings at the University of Cincinnati. These experiments involve treating water samples contaminated with persistent pollutants like bacterial toxins and pesticides. The successful experiment achieved up to 80% reduction in toxins with the integration of nanofiltration membranes, enhancing pollutant retention and minimizing residual toxicity. Although scalability remains a challenge, this approach offers a sustainable pathway for industrial-scale water treatment. Additionally, nanotechnology research led by scientists in India focused on nanostructured materials including carbon nanotubes and graphene for targeted contaminant removal. Nanotechnology experiments were conducted in both laboratories and real-world field settings. In rural India, arsenic-specific nanomaterials were deployed to purify drinking water, successfully reducing arsenic levels to or below World Health Organization safety thresholds. This technology provided safe drinking water to millions of arsenic-affected areas. Additionally, nanostructured filters were tested in regions impacted by agricultural runoff, significantly reducing pesticide concentrations to safe levels. These filters benefited over 7.5 million people globally, demonstrating the practicality of nanotechnology for addressing water quality challenges. These experiments highlight the measurable impact of these approaches, with photocatalysis addressing persistent toxins in controlled environments and nanotechnology showing success in improving water access for underserved populations.
Another way this is being done is through bioremediation, which is a natural process that employs microorganisms to eliminate pollutants from water and soil. This solution relies on the natural ability of bacteria, algae, and other microorganisms to break down pollutants at the molecular level. In advanced wastewater treatment plants, nitrifying bacteria such as Nitrosomonas and nitrobacteria are used to convert harmful nitrogen compounds such as ammonia into less harmful substances like nitrates. Additionally, denitrifying bacteria are used to convert these nitrates into nitrogen gas, which is then harmlessly released back into the atmosphere. This microbial action at the cellular level is crucial in reducing the pollution load and improving water quality.
Moving forward into the future, the authors proposed future experiments where both nitrifying and denitrifying microorganisms would be used simultaneously alongside other biological processes to achieve total nitrogen removal in a single system. They concluded that whilst conducting this experiment, identifying any problems along the way and updating the parameters could create an integrated system that would successfully treat wastewater with multiple pollutants. These experiments could include the use of phosphate-accumulating bacteria for nutrient removal alongside nitrogen removal. However, some potential challenges and concerns of conducting these experiments could include, firstly, carbon source limitation. Denitrifying bacteria require organic carbon to reduce nitrates to nitrate gas. Some wastewater streams may lack sufficient carbon, requiring external carbon sublimation, which could make the process less cost-effective and efficient. Secondly, unpredictable microbial dynamics. Future experiments will need to focus on stabilizing microbial populations as they fluctuate easily, which could lead to inconsistent treatment performance. And lastly, process monitoring and control. As seen from past experiments, microbial populations may fluctuate and without input would result in inconsistent treatment performance. Future experiments will absolutely require real-time monitoring to make sure optimal conditions are maintained and the highest amount of waste is treated.
Finally, it is through the ever-improving detection monitoring of harmful microbial bacteria and pollutants in water sources which is helping to transform more and more wastewater into usable, even drinkable water for current and future generations. Over time, we hope to see the support and awareness of these revolutionary projects through biochemical solutions be valued even more highly, all in the aim to improve the longevity of our species and the species we share the planet with.